Hello everyone, I'm Dr. Marua Abu Risha from the Research Center Borstel in Germany. On behalf of the co-authors, I will be sharing our last publication in Allergy, in which we defined a new method to identify virally trainable genes. Humans are exposed to environmental exposure throughout their life. It has been shown that environmental exposure can affect DNA methylation. This led us to question whether repeated viral infections can reprogram the epithelium. It's still uncertain if such reprogramming would enhance or diminish the response. So to address this, we wanted to use specific design, where we infect the epithelial cells 5 times with human rhinovirus, with two other factors such as duration and multiplicity of infection. But this full factorial design is complex and will involve many samples. However, by using mathematical modeling, such as DOE, we were able to reduce this design, and I encourage everyone to read the paper for more details about this design. Based on the experimental design, DNA and RNA samples were isolated for each infection. A specific mix model was then used to analyze the changes in both DNA methylation sites and RNA expression. We have developed a new method to identify genes that exhibit linearity with infections, and by linearity we mean a steady increase or decrease in methylation and expression. And by using this method, we identified 77 virally trainable genes, and we investigated their pathways and roles within a network. These are the results of one of the trainable genes, S100A8. It has an important role in the danger signaling pathway. As shown here, the expression level is increasing with more infections, and each infection alters the methylation of its CBG sites. However, only one CBG site displayed a linear correlation with number of infections, and at the same time shift the methylation status from partially methylated to unmethylated. As you can see here, the methylation is lowered with more infections. Interestingly, the cohort data showed an increase in methylation with age for the CBG site, but the level are lower in asthmatics compared to the control, indicating that something such as viral infections may demethylated this site as proposed by our tissue culture model. And with that, I encourage you again to read our paper for more details. I would like to extend my gratitude to our partners and co-authors in the Alliance cohort and the Research Center Borstel. We thank the DZL for funding this project and you for listening. Thank you for your attention.